This video will provide detailed protocols for in vitro shape experiments to determine the secondary structure of RNA sequences of interest in the presence of an RNA-targeting small molecule.
In vitro shape is a cost-effective method to understand the dynamics of each nucleotide on a middle-sized RNA element, which is usually less than 200 nucleotides.
After preparing the RNA template as described in the manuscript, radiolabel reverse transcription primers by adding gamma-32P ATP, reverse transcription primer, 10x PNK reaction buffer, polynucleotide kinase, and RNase-free water in a 1.7 milliliter microcentrifuge tube in a total volume of 20 microliters. Then incubate at 37 degrees Celsius for 1 hour. Heat and activate the samples for 20 minutes at 65 degrees Celsius, and then filter the samples in a desalting column and centrifuge at 1000 times G. Add 25 microliters of 2x TBE urea sample buffer and mix. Load these labeled products into a 10% acrylamide gel and make sure to avoid bleeding the radioactivity into the top reservoirs. Run the gel at 20 watts for 1 hour. After the run, remove the glass plates of the gel cassette. Lay the gel on a piece of plastic wrap. Fold the plastic wrap onto the top of the gel to make an airtight sandwich. Place the gel sandwich on a calcium tungstate phosphor screen and expose with an imaging instrument. Image the gel again with regular light to know where the edges of the gel are. Use an image processing software to overlay the two images, and overlay the light.